plant sources of anti-cancer agents are plants, the derivatives of which have been shown to be usable for the treatment or prevention of cancer in humans. Background: in the 1950s, scientists began systematically examining natural organisms as a source of useful anti-cancer substances. It has recently been argued that the use of natural products has been the single most successful strategy in the discovery of novel medicines. Plants need to defend themselves from attack by microorganisms, in particular fungi, and they do this by producing antifungal chemicals that are toxic to fungi. Because fungal and human cells are similar at a biochemical level, it is often the case that chemical compounds intended for plant defense have an inhibitory effect on human cells, including human cancer cells. Those plant chemicals that are selectively more toxic to cancer cells than normal cells have been discovered in screening programs and developed as chemotherapy drugs. Research and development process. Some plants that indicate potential as an anti-cancer agent in laboratory-based in vitro research, for example, Typhonium flagelliform and Murraya knightii, are currently being studied. There can be many years between promising laboratory work and the availability of an effective anti-cancer drug. Monroe Elliott Wald discovered anti-cancer properties in Comptotheca in 1958, but it was not until 1996, after further research and rounds of clinical trials, that topotocin, a synthetic derivative of a chemical in the plant, was approved for use by the U.S. Food and Drug Administration. Plants. Comptotheca acuminata. The cancer treatment drug topotocin is a synthetic chemical compound similar in chemical structure to comptothecin, which is found in extracts of Comptotheca, happy tree. Catharanthus rosas vinca alkaloids were originally manufactured by extracting them from Catharanthus, Madagascar periwinkle. Pedophyllum spp.two. Chemotherapy drugs, etoposide and tuniposide, are synthetic chemical compounds similar in chemical structure to the toxin podophyllitoxin, which is found in Pedophyllum peltatum, May apple. Taxus brevifolia. Chemicals extracted from clippings of Taxus brevifolia, Pacific U, have been used as the basis for two chemotherapy drugs, docetaxel and paclitaxel. Euphobia P+ contains ingenol mebutate, Picato, which is used to treat skin cancer. Maintenance ovitus trastuzumab. Cadsilla is an antibody conjugated to a synthetic derivative of the cytotoxic principle of the Ethiopian plant Maintenance ovitus. It used to treat breast cancer. See also: herbal medicine, experimental cancer treatments, chemotherapy regimens, National Comprehensive Cancer Network, alternative cancer treatments. References. External links. Phytotherapy research. Apigenin in cancer chemoprevention.